everyone. Welcome to today's class. I am Professor Wang. Today, we are going to talk about a major topic in our body, DNA structure and functions. Behind this, we are going to perform a practical experiment, fluid DNA clue extraction test. We will extract it from fluid using simple things from your kitchen, so it's very easy to do by yourself at home. So it's going to be fun. Let's get started. First, let's start with a big question. Why do you look like your parents? Maybe you have your dad's eyes or your mom's hair. This is heredity. It's how traits pass from parents to kids and inherit through the transmission of genes. So, the heredity is referred to the process by which offspring inherit characteristics from their parents through the transmission of genes. And genetics is the scientific discipline that studies this phenomenon. It's now known that most life forms on Earth use DNA as their genetic material. So, the science behind this is genetics. Genetics is the science we use to study this. Everything we are, our physical trait, our health risk, is all written in a code. So, today we are going to open the book where the code is written. So, what does the DNA look like? In 1953, Watson and Crick found the answer and gave us the most famous shape in biology, the double helix. The double helix is the storage gene information place. Most DNA in our body exists in the big form. The big form is a right-hand form. It's a right-hand helix. Now, use your finger hand. Point your thumb up and curl your fingers. The color-wise turn is the direction of life. So, the DNA composed of multiple nucleotides, nitrogenous base, also called nucleobase, A, T, C, G, and deoxyribose, and the phosphate group. And the other, the one completed turn contains 10 base pairs of the nucleotide. Let's move to the next slide. The DNA and RNA. The DNA and the RNA is composed by nucleotides. Many nucleotides composed to the nucleic acid, and the nucleic acid composed to the DNA and the RNA. So in this figure, we can see the DNA called deoxyribonucleic acid composed of the pentose deoxyribose and the base and the phosphate. The base is ATCG. It's double helix, complementary pairing, and containing genetic information here. The other is RNA. RNA is ribonucleic acid. It's pentose or ribose, base, and the phosphate. AUCG is a single strand. The RNA is different with DNA. RNA is single strand and the DNA is double strand. So the RNA is a ribose here, such like the mRNA, RNA, and the tRNA. So let me further to see the DNA structure. The DNA of the base include the purine and the pyrimidine. The purine is adenine, A, guanine, G, and the pyrimidine is thymine, T, and the cytosine, C. This base, A combined with T and C combined to G by hydrogen bond. So these bases are linked by hydrogen bond. Okay, let me see what is gene. The gene is the segment of DNA with genetic information, and functionally is the basic unit that controls traits. The genetic information carried in gene can be expressed through protein synthesis, lead to difference in the phenotype trait of individuals. In our body, how many genes we have? In this slide, we know humans have about 20,000 to 25,000 genes. So, how does DNA work? It uses the gene code. A gene is a small piece of DNA that tells your body how to make a protein. Think of your genome as a big library, and the genes are the books. The code is read in group of 3 letters, called a codon. So in this slide, you can see the genome composed of all genes is the library of genetic information. And in human genome contain about 25,000 genes and can produce about 25,000 types of protein. So it's so huge, so many proteins produced. So in the gene code located on the DNA consists of 4 chemical bases, A, T, C, and G, triplet codon, which code for one specific amino acid. So in this figure, you see TAC to compose the methionine, and the AAA to phenylalanine, and CCA glycine, AGG serine, CCA glycine, and the ACC tryptophan, etc. Yeah. So there are 64 possible combinations in total. So this combination build the proteins that make you are you. Let's see this slide. DNA is actually very, very long. To fit inside a tiny cell, it folds up tightly into chromosomes. Humans have 23 pairs, so 46 in total. In this chromosome, you see the chromosomes are folded by DNA plus the histone to form the nucleosome, and all the nucleosome hold together and close tightly to the chromosome. So DNA to fall to the chromatin and to the chromosome. Okay, so let's see what's the chromosome. The chromatin replicate and condense into visible chromosome only just before cell division. So humans have 46 chromosomes, and 22 pairs are autosomes, and one pair is sex chromosome, is SS or SY. Okay, in this slide, you see the chromosome dominate allele and the recessive expression. You get one gene, one gene, one chromosome, one set from your dad, and one set from your mom. Each gene has its own spatial spot on the chromosome. It's called the locus. In this slide, you see the locus two case study. 2 cases. One case is eyes color, human eyes color. The second case is the flower color. So let's first see the case 1. The 2 chromosomes, one from your father and one from your mother. The mother, father, if the gene is large A, small a, so if father's gene is large A and small a, mother's large A and small a, so in this figure you can see the eyes, brown eyes. Brown eyes is large A, and large A, small a, but also have 1/4 is the blue eyes, small a and small a. So in the case 2, flowers color also is the same principle. So here you see the large R and small r. To form the red flower is large R, small r, and the white flower is small r. So the small r is the recessive trait, and the large R, small r is the dominant trait. Okay, the other example you can see in this slide. Let's look at some examples. Can you roll your thumb? That's a dominant trait. Do you have a V-shaped hairline? That's called a widow's peak, and it's also dominant. Even your blood type, ABO type, is decided by the gene. It's amazing how much is hidden in our cells. So you see in this slide, the widow's peak, also called hairline shape. Widow's peak present is dominant trait. No widow's peak is recessive. And islet, double islet is dominant, and single islet is recessive. Tongue rolling, can roll tongue is dominant, but cannot roll tongue is recessive. Let's see the middle picture. The earlobe attachment. Someone's earlobe is separate. Free earlobe is dominant, and attached earlobe is recessive. Okay. Someone smile very charming. Yeah. He had dimples. If you have dimple, you are very cute. Okay. So dimples present is dominant, consists dominant trait. Okay. And some flexibility. Leave your son. Can you press? Okay. If your son can press, it's recessive. Now reflex is the straight sound is dominant. Okay. And then the other is ABO blood type. ABO blood type also A blood type, B blood type, A and B blood type, or O blood type. This O is the single gene inheritance. Okay. Now let's look at the experiment. We will extract DNA from fluid. Okay. The practice is very easy to perform at home and can do by yourself. So let me go the fluid DNA clue extraction test. First, you will prepare the material. The material is 1/4 kiwi fluid. Kiwi fluid is good fluid, yeah. Or banana. Each one is okay. Then the plastic beaker, or if you have no beaker at home, you can use one cup, and a spoon, and a plastic bag, drink bag. Or the drink bag can replace the funnel, yeah. 50 milliliter centrifuge tube to put the water in. You have not a centrifuge at home, it's never mind. Yeah, you just use 1 cup of water. Yeah, and 15 milliliter centrifuge tube to put the 95% cold ethanol, and the 2.5 milliliter 5N NaCl, and the dish soap. Dish soap is detergent at your home to wash your dish, wash your chopstick, yeah. And the plastic rubber, slide or plastic sheet. This all your material here. And let's go for the procedure. Okay, step 1, sample preparation. You use spoon to take 1/4 kiwi or 1/3 banana to peeled, and press the fluid into the plastic bag. To put it into the plastic bag, and do not add water. Okay, be careful. Do not add water. Just put the kiwi in the plastic and mash the fluid by hand until it reaches a smoothly pour-like consistency. The thinner, the better. So let's move to the step 2. Step 2, dilution and transfer. Pour 50 milliliter water into the plastic bag and in several stages. Put in and mix and wash all the fluid part and the juice into a beaker. And put water and mix and pour out. Yeah, pour out then to wash. And the procedure step 3, lysis. To add 1.25 milliliter of detergent to stir for 3 to 5 minutes to ensure a complete reaction. These beakers don't cell membranes like this figure. Sure. If you have not a dropper to measure the 1.25 milliliter detergent, you can also use a spoon to in this figure show to spoon to for 1.25 milliliter detergent to mix. Then we move to the step 4. To add 2.5 milliliter of 5N NaCl is called saline solution. The NaCl is salt in your kitchen, so it's a common thing. Yeah, so you to add this salt and stir for 3 to 5 minutes to ensure complete reaction. To stir, to mix. And move to the step 5, filtration. After stirring thoroughly, place a drip bag and filter the solution into a paper cup. Procedure. Step 6, sample measurement. Stir the filtrate in the cup until uniform. Take 15 milliliter of the filtrate and pour it into a 50 milliliter test tube. Step 7, alcohol precipitation. Tilt the test tube at a 45-degree angle and slowly, be careful, slowly pour an equal volume, 15 milliliter of 95% ice cold alcohol down the side of the tube. Note, pour very gently, be careful and gently. Try not to disturb or mix the 2 layers of liquid. Okay, until this, can you follow me? Okay, then we shift to the step 8, incubation. Place the test tube in beaker and let it sit undisturbed for 10 to 15 minutes. Ensure it does not tip over. And the relax will usually visible within 3 to 5 minutes. Okay. So you can, after 3 to 5 minutes, you can see the result. A cloudy white cotton-like substance will see is the DNA. DNA will appear at the interface between the alcohol and the water layers. So the result you can see here is the magic moment. Look at the interface. Those white cotton-like fibers, can you see that? That's the DNA of the fluid. Okay, try it. Now, use your laser. See how the beam scatters when it hits the DNA? That's the physical structure interfering with the light. Finally, pick some up and touch it. It's lively and streaming, right? These are the properties of the long-chain polymer. You can see in the picture 1, what happens when you shine a laser line of DNA on DNA. 2, extract the DNA and place it on the slide. Then touch it to feel it. Discussion 1. What is the purpose of adding detergent in this experiment? 2. What is the purpose of adding fine saline solution in the experiment? 3. What is the purpose of adding 95% coethanol at the end of the experiment? 4. Why can DNA be distinguished when illuminated with laser light? 5. Briefly write down your reflection on today's experiment. So, what would happen if we use warm alcohol, not cold alcohol? It's warm alcohol. If we use warm alcohol, what happened? What will be happen? What was the detergent necessary? Why the detergent is so necessary? Okay, congratulations. You are just touched the essential of life. So, it's your turn. Thank you for your attention.